Welcome back to Biology 1620. Today's class will focus on carbon and nitrogen cycles. The key concept to come out of today's lecture will be that living organisms do drive carbon and nitrogen cycles, and that humans are impacting both the carbon and nitrogen cycles. In part one, we're going to talk about cyanobacteria. This group fundamentally changed our planet in two distinct ways. Cyanobacteria is a name for bacteria. There are about 6,000 species of bacteria. Some that are small, such as microcystis. Others are larger, such as oscillatoria and anabina. Some of them are free living in the waters, fresh waters or oceans. Others are found in stromatolites. Some of them even grow in the soil surface. They are associated with algal blooms, but keep in mind they are not algae. They are bacteria. So it's a misnomer when the public calls something like you see in the top left an algal bloom. It's really a cyanobacteria bloom. Cyanobacteria fundamentally changed the world with two enzyme reactions. The first is associated with the carbon cycle, a topic we'll talk about today, and the second is associated with the nitrogen cycle, again a topic that we'll talk about today. It's in the bacteria that we begin to see oxygenic photosynthesis, where CO2 is combined with water to form a carbohydrate and the release of oxygen. And that fundamentally changed the Earth's atmosphere and allowed the rise of the eukaryotes. But you might not realize there was also a second fundamental reaction that takes place, and that's in the nitrogen cycle. Cyanobacteria developed the capacity to take diatomic nitrogen, add energy from photosynthesis, and create ammonia. So essentially they are their own nitrogen factory. And these two fundamental reactions dominate the carbon and nitrogen cycles. If we begin to look at photosynthesis in a little bit more complicated way, we realize that it is the combination of a 1-carbon molecule, CO2, and a 5-carbon molecule, ribulose bisphosphate, to produce two 3-carbon molecules, phosphoglyceric acid. Rubisco, the enzyme here, which catalyzes that reaction, evolved early in the Proterozoic. It was at a time, of course, when there was no oxygen present. But it turns out today, in low CO2 concentrations, that oxygen sensitivity becomes more apparent.